Hello everyone. Before we get started with today's lesson, I wanted us to have a quick look back at a topic we've done previously. So as a starter today, I would like you to answer a couple questions on fertilization. So several processes lead to fertilization, lead up to fertilization in animals and plants. Describe and explain how, in mammals, events following the acrosome reaction prevent more than one sperm fertilizing an egg. If you stop the video here and answer this, please. Okay, so this question means that you actually have to spell any technical terms correctly. So anything that's in italics has to be spelled correctly in order for you to get the mark. So the first thing that happens following the acrosome reaction, if you remember, the acrosome reaction is where the acrosome of the sperm cell swells up, fuses with the sperm cell surface membrane, and then it digests through all the outside layers of the egg, so this follicle layer and the zona pellucida. Then it's able to fuse with the egg cell membrane. Okay, so the sperm has just approached right here and it's finally fusing with the egg cell surface membrane. This fusion causes cortical granules or vesicles. Remember, you have to spell these correctly because they're italicized. Those cortical granules causes them to rush up to the membrane, up to the surface. And you just mentioning the word cortical granules or vesicles in that way can get you a mark. Okay, so these cortical granules are moving towards or fusing with the egg cell surface membrane. You have to have the word membrane there. And those cortical granules or vesicles, if you call them that, are exocytosed. Okay, so exocytosis happens here. They're transported out of the egg here. The idea of the contents of the cortical granules being released, so all the enzymes or the chemicals within them being released into the zona pellucida. They say jelly layer. We want to say a zona pellucida. It's more accurate. Or if you said that this whole thing is called a cortical reaction, you also get that mark. And finally, and this is the mark that I would expect absolutely everyone to get, the idea of the hardening or thickening of the zona pellucida, or the formation of the fertilization membrane. This is the way that we've learned it, thickening of the zona pellucida. Okay, so there's 4 marks there, any 4 of these will do. And then the second question, animals produce haploid gametes by meiosis. Explain how meiosis gives rise to genetic variation in gametes. And again, good exam practice is to highlight any keywords. So we should know what haploid, we should know what meiosis is, we should think about what genetic variation is. You should know these words before you even attempt the question. So stop the video here. Okay, and the answers are, you need to have reference to both independent assortment and crossing over. Both of these things need to be mentioned, and then you can explain each one. So independent assortment gives rise to new combinations of chromosomes. And you can sort of see it here on this diagram. We've got the grey and the red, and if we assume that represents the different parents, the way in which they orientate themselves, we could see that there was one big parent 1, one small parent 2, but it could have equally been that both of the grey ones end up together and both of the red ones end up together. It was random the way it happened here, and this is only 2 chromosomes, and we have 23 pairs, so obviously that would be a lot more complicated. And then the second mark, or the third in this case, crossing over involves swapping of sections of chromatids. They would have accepted chromosomes. If you have put sister chromatids, you do not get this mark. It should be non-sister chromatids, or in a more basic way, just chromatids. But if you said sister, you do not get this mark. You also do not get the mark if you said that it's swapping of genes. It's not. It's creating new combinations of alleles by swapping over sections of the chromosome. Okay, so that's our little starter for today. The topic I would like to cover today is epigenetic control of gene expression in eukaryotes. And I believe this is the last topic we're doing in Unit 3. So we're moving on to Unit 4 next lesson. The learning objectives for today are to describe what is meant by epigenetics, DNA methylation and histone acetylation, and explain how epigenetics can result in organisms with the same genotype, so the same genetic material, but different phenotypes, so different expressed observable characteristics. And before we get into the lesson, let's go through some key terms that you will see again and again in this lesson. So epigenetics overall, the topic for today, is the study of inheritable changes in gene function without changes to DNA-based sequences. So there are actually possibilities where the expression or the phenotype of an organism changes even though the DNA sequence didn't change. So there's not been a mutation, but there is something happening to the DNA. The environment of the DNA changes, and that can affect whether a gene is expressed or not. Genome is all of the DNA in an organism. A methyl group is one carbon bonded to 3 hydrogens. You may remember that carbon should form 4 bonds, and the 4th bond here would bind the methyl group to whatever molecule the methyl group is being bound to. You see later on in the PowerPoint. And finally, histones, proteins that DNA wraps around to form chromatin. Okay, so this is what helps it condense down into a chromosome. Methyl groups and histones are going to come up because they are 2 of the big ways in which we can actually modify DNA in a way where we don't change the DNA sequence, but we change the environment of the gene. So just to remind you what a histone is, you can see here you've got your DNA, and that wraps around this histone. This is a protein molecule, and the DNA wraps tightly around it, and this is kind of what helps it organize into this nice pattern that can eventually condense down into a chromosome. Okay, so things can happen here which affect whether genes are expressed or not. And this is a really interesting field of study. There's been lots of experiments done on mice. In this particular one, you've got an overweight yellow mouse mother, and they've done a few studies. In one of those, they fed a pregnant mother with a diet that was supplemented with compounds rich in those methyl groups that I mentioned earlier. And what happened in those mice is that the offspring was predominantly brown and in good health. There were a few that were more like the mother, but actually most of them were pretty healthy. Whereas if you feed the mother, the pregnant mother, regular mouse food, what they saw is that the mice tend to look like the mother and be in poorer health, so be overweight and be yellow. So this is the route that I would take normally if you just give them normal mouse food. So, and that makes sense. That's what we're used to. We're seeing a young offspring inheriting the parents' characteristics. But if we change the environment of that gene, so if we're adding methyl groups, that somehow affects expression of the genes. Something has happened here, and that's what we're going to look at today. So as I mentioned, epigenetics involves inheritable changes in gene function without actually changing DNA-based sequences. These inheritable changes are caused by changes in the gene's environment, and you can inhibit transcription of those genes by 2 of the things we've already mentioned. So that's methylation of DNA. So increased methylation of DNA will stop transcription, or you can decrease the acetylation of histones. And don't worry too much if this is confusing. I will show you some diagrams in a minute. An interesting thing is, most epigenetic marks like this should be removed during reproduction. So although they can affect gene expression in the organism which has it, most of those do get removed across generations. The mouse example we just saw did not have it removed, so some of them do actually stay. So we'll start with acetylation of histones. Okay, so here we've got a diagram of histone molecules with DNA wrapped around them, and you can see that these histones have these tails sticking out. Okay, histones are proteins, so they are chains of amino acids bound together by peptide bonds, and there are these side groups sticking out. You can add acetyl to these side chains. Okay, so histone molecules have these side branches or tails, and these tails contain the amino acid leucine. Leucine is an amino acid that can have an acetyl group added to it. Okay, so that's the amino acid that's very important in this case. Now, if you have lots of these acetyl groups added, that means that it's really difficult to wrap DNA around the histone. So you actually end up spreading out your DNA. And that means that RNA polymerase, the enzyme that you need to be able to transcribe this section, can access. There is space for it. Okay? If it's wound too tightly, like what we see here, then transcription cannot happen because RNA polymerase cannot get in there. So your protein is never made and the gene is said to be switched off. Okay, so just to see it again in a diagram form, the winding of DNA around histones can be tight or it can be loose. When acetylation of histones is decreased, that means that histones become quite tightly packed. The gene is not accessible to RNA polymerase, and you can see it here. It cannot access, there's not enough space. But when you do add acetyl groups, and think again, what was the amino acid? I'm not going to tell you. Think again, what was the amino acid where the acetyl group is added? If you do add loads of them, then the histones become more loosely packed, and that means that RNA polymerase can access and can transcribe that piece of DNA into mRNA, and from then on obviously you can start the rest of your protein synthesis. So gene is accessible to RNA polymerase, and so gene expression takes place. And this is an interesting image because it kind of summarizes everything we've been looking at. We know that stem cells are undifferentiated, and after a while, after lots of genes are switched off, it becomes a differentiated cell, and we can kind of see it here. So this is the DNA of a stem cell, where clearly there's been some acetylation. We don't actually see the acetyl groups here, but we can see that the histones and the DNA are quite spread out, so the DNA is loosely packed. So that means that RNA polymerase can access and can transcribe any of the genes. In a differentiated cell, clearly there's not been very much acetylation, or the acetyl groups have been removed, and so there's really tight packing, and so RNA polymerase actually cannot access. So, the next section is the methylation of DNA. And if you remember, methyl group is a carbon molecule with 3 hydrogens, and the 4th bond is binding it here. And actually, the base that it binds to is cytosine. So just as the acetyl groups are added to only leucine, the amino acid, methyl groups are only added to the DNA base cytosine. When a methyl group is added, it changes the shape of the cytosine molecule, so RNA polymerase cannot recognize it. It's no longer complementary, so it cannot bind. If RNA polymerase cannot bind, transcription cannot happen, so you cannot make any mRNA. And that changes the expression of genes. So in this case, a gene is repressed. And so if we see an example here, let's pretend that we have a section of DNA here. This is just one side of the DNA double helix. So we've got a gene here that should code for salivary amylase, so an enzyme that you should find in your saliva. And then let's say we've got a gene for hemoglobin here. Okay, so this is molecule found within red blood cells to carry oxygen around the body. Let's assume that we are currently in a cell inside a salivary gland, so we should be able to make salivary amylase there. But obviously, every cell in your body does contain the same DNA as the rest of your body, so it will still have a hemoglobin gene. But in this instance, it doesn't really make sense to make hemoglobin. That's not the function of the cell. So you can see here that cytosines in hemoglobin have been methylated, so a methyl group has been added. That means that when you transcribe, you do transcribe mRNA for salivary amylase, remembering that uracil binds to adenine because thymine does not exist in mRNA. But you cannot transcribe the hemoglobin gene. The RNA polymerase cannot bind here because of these methyl groups. It's the wrong shape now. So we actually make a salivary amylase molecule. We make this enzyme, but we do not make hemoglobin in this case. Okay, so as a summary, what I would like you to do is fill out the following table. So tell me what happens when methylation of DNA is increased, what happens when it's decreased, and how does this affect the rate of transcription? And then what happens when acetylation of histones is increased or decreased, and how that affects the rate of transcription. If you stop the video here. Okay, so when DNA methylation is increased, it means that more methyl groups are added to cytosine. This prevents RNA polymerase from binding to DNA, and that affects the rate of transcription by decreasing it. The gene is not expressed. When DNA methylation is decreased, then methyl groups are not added to cytosine, and so RNA polymerase can bind normally. That increases the rate of transcription because the gene is expressed. What happens when acetylation of histones is increased? That means that more acetyl groups are added to the amino acid leucine. That means that we have loosely packed DNA and histones, And that means that RNA polymerase can access and can transcribe the gene, and so the rate of transcription is increased. When acetylation of histones is decreased, that means that there are no acetyl groups added to leucine, and so DNA is tightly packed around the histones, and that decreases gene expression because RNA polymerase cannot access. And just final summary to give you it in diagrammatic form. If we do not have any acetyl group, so if acetylation is decreased, RNA polymerase cannot bind and the gene is not active. You cannot transcribe that gene. When there is increased acetylation, RNA polymerase can bind and can transcribe mRNA, so the gene becomes active. And the main difficulty that I've seen people have, or students have in the past years, is remembering that it's sort of a flip-flop situation here. For DNA acetylation, decreased acetylation means the gene is not active, whereas it's kind of a flip-flop situation when we talk about methylation. So, when we decrease methylation, the gene is active. Okay, so there are no methyl groups, and that means that you can transcribe mRNA. And hopefully you can see here what I mean. So decreased acetylation means gene is not active, but decreased methylation means that gene is active. So you do actually kind of have to almost visualize what is happening to remember what happens. And then if we have increased methylation, that means that RNA polymerase cannot bind, and so you never make the mRNA, and so you never make the protein. So we say the gene is not active. Okay, so what I would like you to do is to check your knowledge. I'm going to give you a few summary questions and then an exam question. I'm not going to give you the mark scheme for this because I would like you to submit the work by the end of the day today, and then I will have a look at it for you and give you some feedback. Okay, so the questions are: a single genotype of plant was subjected to nutrient deprivation and was left to produce a second generation of plants. Most plants of the second generation were found to have an increased number of methylated genes. Question 1. What would be the effect of methylation on the affected genes? Question 2. Suggest why a single genotype of parental plant was used. So why did they just do one genotype? Question 3. A small number of individuals in the second generation did not show the methylation changes. Explain why this could be. And then fact recall questions. Question 1. Give 2 examples of epigenetic marks. Question 2. How can histone acetylation affect gene expression? So what happens when you add acetyl to histone? And then your exam question, and this is for 3 marks. The color of some types of mice is affected by the diet of the mother when she is pregnant. In an investigation, pregnant mice were separated into 2 groups, A and B. The embryos all had the same genotype that should produce a yellow coat. Dietary supplements were given to Group A, and they gave birth to mice with brown coats. Group B were not given dietary supplements and gave birth to mice with yellow coats. Explain how this could provide evidence for epigenetic modification. So what is it about this scenario that means that it's epigenetics? How do you know that it's not just inheritance from the parents, or that it's not just mutation? Okay, have a go at this and submit the question by the end of the day. Any questions, just let me know. Thank you.